This video will compare the structures and functions of insect fat body cells and vertebrate adipocytes. As discussed in Scene 1, the insect fat body serves a functional metabolic role similar to the vertebrate liver. There are three cell types in the insect fat body. Some early investigators initially termed the fat cells trophocytes, but they resemble in function similar to the adipocytes of brown fat from vertebrate animals. Fat cells of vertebrate animals consist of three adipocyte types, depending on the physiological state of the host animal: white adipocytes, beige adipocytes, and brown adipocytes. White adipocytes are found in hibernating vertebrate animals. Their function is to store fat to sustain the dormant animal through winter hibernation when food is scarce. White adipocytes contain a single, enlarged fat vacuole for lipid storage. The vacuole nearly fills the entire volume of the cell and causes the nucleus to be offset, so there is a reduced peripheral cytoplasm containing little endoplasmic reticulum, Golgi bodies, and mitochondria. By contrast, beige adipocytes are found in animals awakening from hibernation. They are intermediate in structure between white and brown adipocytes. They are hormonally activated white adipocytes that are undergoing browning to become metabolically active brown adipocytes in active vertebrate animals. Finally, in active vertebrate animals, brown fat adipocytes contain numerous fat storage vacuoles with abundant endoplasmic reticulum, many Golgi bodies, and mitochondria. The abundance of these intracellular organelles indicate brown fat is metabolically active. In fact, vertebrate brown fat is a thermogenic tissue and is responsive to metabolism-stimulating hormones that increase heat production in dormant vertebrate animals to terminate hibernation. Insects' fat body adipocytes resemble vertebrate brown fat and contain multiple fat vacuoles throughout the cytoplasm, along with abundant endoplasmic reticulum and mitochondria, indicating active biosynthetic capacity. Insect fat bodies often have three cell types: adipocytes, urocytes, and mycetocytes. Fat body adipocytes are present in all insect orders, with urocytes and mycetocytes limited to selected orders. In a typical lobe of cockroach fat body, the outermost cells exposed to the hemolymph are usually metabolically active adipocytes. More centrally in the lobe are urocytes with uric acid stores in close association with mycetocytes containing bacteroids when present. This close arrangement of urate stores with symbiotic bacteroids suggests uric acid nitrogen may be recycled by the bacteroids for biosynthetic processes. Finally, one of the major roles for the fat body is to facilitate the absorption of dietary carbohydrate. Dietary carbohydrates consist of mainly glucose. Digestion-derived glucose passes from the crop into the gastric ceca for absorption into the body. Glucose concentrations are normally low in insect hemolymph, so a glucose diffusion gradient exists from the gut to the hemolymph. The low hemolymph glucose allows dietary glucose to pass from the cecum into the hemolymph by passive diffusion. Thus, active transport is unnecessary for glucose absorption. Hemolymph glucose is rapidly taken up by the surrounding fat body adipocytes, where it is converted to trehalose, a chemically inert disaccharide comprised of two glucose subunits linked at their reducing carbons. In this manner, hemolymph trehalose serves as a non-reactive carrier molecule for glucose transport to insect tissues. In summary, the fat body conversion of dietary glucose to trehalose, then back to glucose, has several benefits for insects. Insects are active animals that require a readily available source of metabolites for muscular energy. However, insects have an inefficient open circulatory system which does not readily direct hemolymph metabolites to the muscles to support this energy demand. Like vertebrate blood, insect hemolymph must stay within the physiological limits for properties such as osmolarity and metabolite composition. Furthermore, by using trehalose as a mobile source for glucose, 2 glucose molecules are delivered to the muscle cells by a single trehalose molecule. Trehalose delivers twice the energy to the muscle cells, but with half the chemical effects of free glucose on hemolymph composition. In closing, it should also be noted that no additional energy is required for the uptake, transport, and chemical conversion of glucose to trehalose. The insect fat body is a dispersed, multifunctional, hormonally sensitive tissue that responds to the unique morphological and physiological demands of insect growth, metamorphosis, and reproduction. This tissue has helped insects to become one of the most varied, prolific, and successful forms of life.